Olaparib is an anti-tumor PARP inhibitor drug that competes with NAD+ and causes double-strand breaks that cannot be repaired in tumor cells with inactive homologous repair pathways. Homologous repair mutations such as BRCA1 and 2 are common in ovarian and breast cancers. Olaparib is a small molecule with a molecular weight of approximately 434 daltons. It is moderately lipophilic due to aromatic and cyclopropyl rings, and is mostly unionized at physiological pH. It is structurally similar to the nicotinamide portion of NAD+. Olaparib can therefore cross cell membranes due to its hydrophobicity, and therefore it can be taken orally in a pill or capsule. Olaparib acts on PARP. Poly-ADP ribose polymerases, PARPs, are key enzymes in recognizing and flagging single-strand DNA breaks for repair. PARPs contain three key functional regions. The DNA binding domain binds nicked DNA via zinc finger motif. The catalytic domain synthesizes poly-ADP ribose polymers, PAR polymers, from NAD+. The NAD+ binding site is within the domain. The auto-modification domain accepts PAR chains from the catalytic domain and regulates dissociation from the DNA due to negative charges on PAR chains and DNA backbone repulsing each other. Normal action of PARP. A single-strand break occurs and is detected rapidly by the zinc fingers on a PARP, tightly binding to the damaged DNA. DNA binding activates the catalytic domain of PARP and allows NAD+ access to its binding site. NAD+ is cleaved into nicotinamide, which is released from the enzyme, and ADP ribose, which is retained. ADP ribose is polymerized onto target proteins, such as histones, excision repair proteins, and the self-modification region. Autopyrylation causes dissociation from DNA. Histone pyrylation causes chromatin remodeling, and ER protein pyrylation causes activation and DNA repair. Once PARP is dissociated, excision repair can take place. Olaparib competes with NAD+ for the PARP binding in the nicotinamide binding pocket. This inhibits autopyrylation and PARP dissociation. This results in formation of strongly bound PARP DNA complexes. During the next S phase, replication forks collide with PARP. The forks stall and signal replication stress. If the stalled forks are not stabilized, one NADSBs form. One NADSB is highly cytotoxic and can only be repaired by homologous repair. In healthy cells, they have active homologous repair pathways and can fix the DSB. Tumorous cells with an active homologous repair enzyme such as BRCA1 and 2 mutants cannot fix the DSB. They will undergo apoptosis due to buildup of DSBs and signaling via ATR, CHK1, ATM, or P53. Inactivity of HR enzymes is what allows Olaparib to target tumors. This is called synthetic lethality. Stephen Jackson's group in Cambridge realized the therapeutic potential of PARP inhibitors in cancers that lacked HR pathways by synthetic lethality. The group used NAD+ as a starting structure and generated drugs that mimic NAD+'s shape. The drug put to trial was Olaparib. Olaparib, sold as Lynparza by AstraZeneca, was the first PARP inhibitor approved by regulatory authorities for treatment of BRCA mutant ovarian cancer. Most side effects occur in tissues that are rapidly dividing. These tissues divide slower with Olaparib due to the time taken for HR mechanisms to repair DSBs. Examples include hematopoietic cells in bone marrow. This causes a decrease in blood cell synthesis, causes anemia, neutropenia, lymphopenia, which weakens the immune system. Another example is rapidly renewing mucosa. Low mucus levels in the gastrointestinal tract causes nausea, vomiting, and diarrhea. Olaparib is delivered orally due to its ability to pass to the intestine unchanged and fast absorption due to small uncharged nature. It has a large volume of distribution, showing large tissue and tumor penetration, although a poor blood-brain barrier penetration due to efflux transporters. It is hepatically and renally metabolized, with moderate first-pass metabolism. With a half-life of 12 hours, steady state is reached in 3 days, following twice-daily dosing. Excretion is mostly fecal. There's many possible ways for a tumor to gain resistance to Olaparib. The two most common are BRCA reversal mutations, which reactivate homologous repair pathways and stop synthetic lethality, and loss of fork-degrading nucleases MRA11 and upregulation of fork protection factors such as RAD51. This causes less double-stranded breaks.